हेलो फ्रेंड्स। एज पर आवर डिस्कशन प्लान इन एनसीईआरटी आज हम एक टॉपिक डिस्कस कर रहे हैं दैट इज नेम्ड एज phosphorylation. Foremost, we need to understand the meaning of phosphorylation. It is the process in which phosphate is added in a compound to form high energy molecule like ATP. Like if someone added ADP with an inorganic phosphate, it will convert it into ATP, that is called as phosphorylation. The formation of ADP and PI addition to form high energy compound, that is ATP, requires three type of conditions. First is, if we discuss that how ATP will form, we can have three mechanisms. There are three mechanisms for doing this phosphorylation. First is called as substrate level phosphorylation. Second is called as photophosphorylation. And the third one is called as oxidative phosphorylation. If I talk in this way, like these phosphorylations are very important concepts, we can say that substrate level phosphorylation takes place inside cytoplasm. Photophosphorylation takes place inside chloroplast or chromoplast, while oxidative phosphorylation takes place inside mitochondria. These are the organelles or cellular part in which these phosphorylations takes place. If I talk in this way, substrate level phosphorylation common in all organisms, photophosphorylation in green plants, while is oxidative phosphorylation in all eukaryotes. Let me introduce what are these three things. In first process, that is called as SLP, second is called as photophosphorylation, and third is called as oxidative phosphorylation. If I talk in SLP manner, substrate level phosphorylation, what will occur that some high energy compounds present, and this high energy compound transfer its energy, it produces some bond energy, and with the help of this bond energy, ADP can phosphorylate, in presence of inorganic phosphate into ATP. While you talk in second activity, that is photophosphorylation, it means that in chloroplast, like if you talk thylakoid membrane, you can understand it in clear way. This is thylakoid membrane, and these are actually photosystems. This is photosystem 2, this is cytochrome B6F, and this is photosystem 1. During this transfer of electron from photosystem 2 to 1, when water is broken here, you can say that proton collected in this place, O2 released in environment, and electron transported in this. This leads to the formation of NADP H2 via NADP plus NADPH plus H plus will form here, and during this transfer protons gets collected in this lumen. When these protons move away via ATPase, they'll produce ATP again. As we need to understand that this water breakdown is facilitated by sun rays, that is why this process is called as photophosphorylation. If we move to the third concept, that is oxidative phosphorylation, we need to understand the physiological differentiation takes place inside mitochondria. In mitochondria, inner membrane contain different complexes, at least 4 of them involved in transfer of protons from here to matrix, that is inner part, to perimitochondrial space. From perimitochondrial space, these protons gets collected in large concentration and created a proton motive force. After some time, these protons return into matrix again. When there are 2 protons returned, again the formation of ATP takes place inside the matrix of mitochondria. As this require the energy from oxidation of NADPH or NADH, that is why this process is called as oxidative phosphorylation. Let me elaborate it for you that this electron transport is started by NADH or NADPH when it oxidizes into NAD plus or NADP plus. So this process is oxidation and it drives the whole process. That is why it is called as oxidative phosphorylation. That is all for today.